Our final speaker for the evening is Dr. Shakeri from the University of Toronto. His research interest integrates advanced biomaterials, microfabrication, and surface engineering to develop biomimetic, tissue models and diagnostic devices. Try saying that 5 times fast. Dr. Shakeri notes that he was always fascinated by the idea of building organs outside of the body, almost like something out of a science fiction movie. The ability to observe cells in action within miniature bioengineered organs is incredible. It can help us study disease more closely, uncover innovative treatments, and develop solutions with a lasting impact for human health. Here is Dr. Shakeri.
Hi everyone, my name is Amit and I'm really excited to share my research on the heart's hidden shield against cancer. Cancer has been the second leading cause of death worldwide, but nowadays, thanks to advances in medicine, many types of cancers can be treated effectively. But the treatment gets challenging when cancer spreads in the body, a process called metastasis. When this happens, the cancer cells dislodge from the initial tumor, circulate within the bloodstream, and invade other organs. They can spread to the stomach, for example, liver, and other organs. But here's something strange. Cancer almost never spreads to the heart. Think about it. Has anyone ever heard of heart cancer? I really hope not. So this is a very rare phenomenon. But why is the heart so resistant to cancer? This is a question I'm investigating in the radiologics lab at the University of Toronto by modeling cancer metastasis outside of the body on a chip. Now, to model cancer metastasis, the first step is to create a porous tube so that the cancer cells can get into and spread. Next, we place this tube in a platform with 5 chambers, and the chambers are only connected together through the tube. Then we grow a mini version of human organs inside these chambers, for example, heart, liver, and breast cancer. Now, to stimulate blood flow, we place the entire platform on a rocker with gentle tilting motion, causing the media to go back and forth through the device. To create the porous tube, we use 3D printing with a coaxial needle, which is basically 2 needles, one into the other one. Now, through the outer needle, we perfuse a biocompatible polymer called POMAC, but in the shape of granules or microparticles to get the porosity. Through the inner needle, we perfuse a temporary gel just to keep the tube open during the printing, and it will be washed away after. As the materials get out of the needle, we shine it with UV flashlight to basically solidify it. I coined these tubes permeatubes because they can mimic the permeability of blood vessels. Here you can see how we use a 3D printer to precisely print these tubes. And here is how the final version of the device looks like. Looking at the permeatubes at higher magnification, these SEM images show the topography and cross-sectional of the tubes. You'll notice there are 2 types of porosities. There are bigger pores with a size of around 40 micrometers, and inside them there are smaller pores with a size of 2 to 4 micrometers. So we started off by studying liver metastasis. We first grow the liver tissue on a chamber on the right, and then once the cells are established, we introduce a highly aggressive breast cancer cells into the middle well. Here you can see the permeatube in blue and cancer cells in green, how they want to enter the porosity of the tube to spread. Next, we track the cancer cell migration over time. So we use a fluorescent microscope where only the cancer cells and the tube were visible in green. Day 0, right after introducing the cancer cells, you can see all the cells are in the middle chamber as expected. On day 6, you can see a few of them have already migrated to other chambers. And by day 10, they're fully migrated across the platform. Next, we repeated the same experiment, but this time we included the heart tissue. Here you can see a video of the heart that we created out of stem cells. You can see how nicely it's beating. This little heart is only 2 millimeter by 2 millimeter its size. Apparently, it's too small for a Valentine gift, as my wife said. But anyways, we tracked the cancer metastasis in this platform as well. And on day 0, as expected, there was no metastasis. Day 6, we still didn't see any metastasis. And even by day 10, there were barely any cells metastasized in other chambers. So why is the heart so resilient against cancer? One reason could be attributed to the proteins that are secreted by the heart, which can prevent cancer cell migration. We, in fact, are going to dig deeper into the tissues in our platform to analyze such proteins. Who knows, maybe one day we can come up with a drug that is inspired by the heart environment to combat cancer. After all, the heart might not just beat for us, but it might also hold the key to fighting cancer. Thank you for your attention.